Hi, everybody. This is just a quick overview of some things that I noticed from the first couple of labs that I want to highlight just as we go forward. So I'm going to jump into the cardiovascular system, start with this lab first, and go over to histology and look at some blood smears. Something that I noticed is that there was just a little bit of confusion in identifying some of the white blood cells. So I want to take a moment just to go back over that. So before I do, here's just the drop-down menu. But in your textbook, section 18.3C, it has the leukocytes section, and it has this table which then gives you all the characteristics of your different leukocytes. So under here, it'll list the type, and then it will tell you the characteristics of its nucleus, what kind of color the granule stand, if it's a granulocyte or an agranulocyte. And so this is really helpful in looking at those physical characteristics and trying to see, okay, well, what am I trying to look at to identify here? So starting with our neutrophils. Yeah, we're good with the erythrocytes. Everybody got those pretty well. It was really the white blood cells. So, starting with the neutrophil. Neutrophil is one of the granulocytes, and so it has this kind of speckled cytosol where the stain is absorbing into those granules. What's really noticeable or kind of identifying about the neutrophil is that it still has a fairly pale cytosol. So, even though it is a granulocyte and you can see those, you can still really clearly see through the cytosol where it's not stained with the granules, and its nucleus has this multilobular shape. So there's four very distinct lobes to its nucleus, and it has these thin stalks kind of connecting them. And so those things are characteristic for a neutrophil. You're going to be easily able to see and identify the nucleus. It's going to have large lobes to it, a fairly pale cytosol with those staining granules. So that's going to be your neutrophil. It's very common to confuse neutrophils and eosinophils, and it's kind of easy to see why. They have similar kind of nuclei where you have these large regions, but you notice that it's a lot more granules, a lot harder to see through the cytosol. The cytosol of an eosinophil isn't as clear as it is in a nucleus. It's not as pale, so you really can't see through it as easily. And the nucleus, if we could see it clearly, is only really two lobes. Like this stalk right here is not as thin on an eosinophil as it is on a neutrophil. So I'm going to go back and forth between those two for just a moment. This is your eosinophil and then your neutrophil. Look how pale that is versus how stained and non-clear. You can't see through the cytosol of an eosinophil. So that's kind of an easy way to tell the difference between those two. Next one is the basophil, and again, it's common to confuse eosinophils and basophils. And so again, you can see it's a really dark nucleus. You can't see through the side or dark cytosol. You cannot see through it at all. There are some kind of clearer spots that indicate some granule staining. Really not a lot of distinct features on the nucleus that we can see. And so this is kind of characteristic of your basophils, is that they're going to have this really deep blue, almost violet color staining to them. You're not really going to be able to see through the cytosol. And if you could see the nucleus, it is kind of similar to an eosinophil in that it has two lobes to it. But again, just jumping back and forth between those two, the eosinophil you'll see has this much more reddish pink stain to it, whereas basophils are going to be that really deep blue violet. So what I'd get as well is basophils and lymphocytes. So this is a basophil. This is a lymphocyte. So lymphocytes are one of the agranulocytes, meaning that their granules are so small we can't see them. You can see though this large structure right here, this isn't the cytosol, this is the nucleus. The nucleus of a lymphocyte pretty much takes up the entire cell. All of the cytosol we can see is this thin little clear circle kind of around it. And so the cytosol that is visible in a lymphocyte is fairly clear. It doesn't really absorb a lot of stain. The dominant feature is this large, very circular, very round nucleus. And so sometimes people will confuse lymphocytes and basophils because they see that large nucleus and they think, oh, it's just taking up all the structure. Whereas a basophil, it has the granule staining and very dark cytosol, still stained cytosol. Whereas with your lymphocyte, the cytosol that you can see is pretty clear. The last of the leukocytes, your monocyte. Again, it's an agranulocyte. You are seeing some staining in here. These are non-specific granules. Those are probably other cellular structures, not an actual granulocyte. What's really characteristic for your monocyte is this large lima bean, butterbean shaped nucleus, kidney bean, whatever you want to call it, or some kind of bean shaped nucleus, not a chickpea. Uh, but it has a really dark nucleus, very large, kind of pushed to one side, and then this really kind of clear cytosol. So it's not a lymphocyte. A lymphocyte would be a perfectly round, very large circular nucleus. It has this indentation, which is really characteristic for a monocyte. So that's a nice, easy kind of way, again, using that chart in 18.C3. It has kind of stylized images and then these images from APR as well to help you identify those white blood cells. And then these kind of written characteristics can be helpful. So if you're doing flashcards, it might be nice to add that to there as you're preparing for your lab practical for next week. The other one I wanted to go into is our endocrine system, and just review again some of the histology of our structures. So, okay, yeah, we got a pituitary. Let's start with that. Sure. So, if we're looking at the pituitary gland, again, it's very characteristic of its shape. It has this large, round, bulbous end, a thin stalk connecting it up to the hypothalamus, more superior. When we're looking at the pituitary gland, we have to remember that there is both the anterior region and the posterior region. The posterior region is also called the neural region. And if you look at this, I'm going to have to click off of it, sorry. There we go. If you look at how pale this is, part of that is because it's more neurons, and neurons aren't going to absorb as much stain as some of these larger cellular structures that are in the anterior pituitary. The anterior pituitary is going to be producing more hormones. It has more cell structures, whereas the posterior is that neural portion, and the nervous portion just doesn't absorb as much stain. So that's kind of a quick way to identify posterior neural versus anterior more cellular. The thyroid gland. So this is this kind of cell structure. This cells ringing this large central fluid packet. This is very characteristic of your thyroid gland because you're producing thyroid hormone inside of these fluid cells, these vacuoles, if you want to call them that. And so the cells that ring around there, you have the really numerous ones are going to be the follicular cells, and then you have much fewer parafollicular cells. That's kind of, and you notice how the parafollicular cells, their shapes are much more oblong, much more kind of distorted, whereas the follicular cells tend to be much more round, much more uniform in shape, and they're surrounding right around this colloid fluid, so that way they can be involved in that process of thyroid hormone synthesis. Let's see, big thing is the adrenal gland, the suprarenal gland, however you want to call that. Let's look at this image is good. Okay, here we go. So the way that I always kind of remember it, sometimes it's easiest to start, is that down here at the bottom, this is the most inner, so this is the medulla in the middle. And then I can see these three distinct regions, one here, and then this one up here. You notice how their color changes? Colors, the staining changes because there's different cell structures that are in there absorbing different types of stain at different rates. And so you can kind of see the distinct changes in that color are going to indicate the changes in those regions. And this outer portion here, there's really not a lot of stain absorbing, and it's because it's mostly connective tissue. That's the capsule of the adrenal gland. And then you have, I always just remember GFR, glomerulosa, fasciculata, reticularis. And let's see if we can go in a little closer. Here we go, great. So the terms glomerulosa and fasciculata, they actually mean things. So you're going to see when you get into the urinary system soon, glomerulosa, glomerular is the shape. Same thing with fascicle, it's referring to a shape. So if something is glomerular, it means that it's kind of roundish. And so you can already see some of these round structures that are in here. And so that's the zona glomerulosa. Oh, of course it's not. There we go. So you can see these kind of round glomerular structures. And so that's why it gets its name of the zona glomerulosa, because it has those kind of things in there. Below it, you can already hopefully see that there's these more up and right, up down vertical structures, more stick-like. And so a fascicle, a fasciculata, see if I can get just one. I don't think so. Oh, there we go. Great. So you can see just these individual fascicles. That tends to be to refer to things that are kind of like stick-like in structure. So instead of glomerular round, a fascicle is going to be more like an upright, lengthy structure. And so when you're looking at some of these zones, make sure that you're looking for the different changes in color. the color gradients. And if you can identify, if you're zoomed in close enough, look for the shapes as well to be able to help you identify transitions in different regions. Okay, I hope that helped. If you have any other questions, please reach out.